Hello, my name is Mateusz Chilinski and I'm a member of Professor Plewczyński's laboratory at Faculty of Mathematics and Information Science at Warsaw University of Technology, as well as Center of New Technologies at the University of Warsaw, both in Warsaw, both in Poland. Today I'll be presenting a poster entitled Enhanced Performance of Gene Expression Predictive Models with Protein-Mediated Spatial Chromatin Interactions. If you're interested a little more than just the poster, feel free to check out our preprint. It's already on bioRxiv. So it's of course explained in more detail in both the manuscript and the supplementary methods. Okay. So the whole motivation behind the study is that with the improvement in the artificial intelligence in the machine learning models, we were getting more and more models that go from the sequence and predict the gene expression. The two most popular, with the highest predictive score are Expecto, which uses convolutional neural networks, or Informer, which uses transformer architecture. We took under microscope Expecto mostly because it goes from the sequence to 2002 features that are different kind of ChIP-seq, ATAC-6, and other epigenetic features. And we ask ourselves the question, as we mostly deal with spatial genomics. Okay, 2002 features is, well, as you can see, quite a lot of features. And what if we extend it by adding some special features? In our case, it will be in form of the heat maps from various experiments of ChIAPET. We had more than 20 cohesin ChIAPETs, as well as 4 CTCF and 4 RNA-pool2 ChIAPETs. And we applied ChIAPipe, or it was already applied by other consortiums, and we took those heat maps and we simply added them into the Expecto model. Let me show you how it worked. So as the input, we had the spatial heat map, and we took the regions that are spatially close to TSS, but they are not close in linear way, because the original Expecto model, you can see here, is taking 20 kb upstream and downstream from TSS, and we are taking the ones that, as you can see here, are for example, very, very far away, right? But they're still close in spatially sense. So for example, this is like, I don't know, 100 kb, this one might be additional 100 kb, so this is like 200 kb apart, but it's close in spatial sense, so we're still taking that sequence. And then we apply the Expecto model, which uses deep neural network, the CNNs that I mentioned before. And we are getting tensors for each of the genes with those special features. There's also special feature transformation in case of the baseline model, and I refer you to the original paper if you're interested more. Based on those tensors, which are predicted features, we construct tissue-specific decision trees that predict tissue-specific expression. Going to the results, we've created, we are comparing ourselves with the baseline model, so the one without spatial expression, and as you can see, we conducted quite a lot of experiments because the algorithm that we use is not deterministic, because it's like decision trees from ECCB. And we conducted each of the experiments from each of the ChIAPET experiment 1,000 times. And here we have, as you can see, the average improvement on the cohesin data set, which is composed of more than 20 cell lines, the CTCF and RNA-pool2, which has 4 in each case. You can see that even in the average scenario, we are improvement. However, the improvement is the highest in the RNA pool 2 case, which is quite obvious if you think about the biological interpretation of the results. And the increase is even by 0.05 in case of RNA pool 2 GM12878. It looks quite small. However, we need to remember that the original model, both expecto and former, are explaining the prediction quite well. In case of expecto, it was around 0.8, so we're explaining around like 20% of, let's say, of the unknown using just adding those spatial features. Also, the improvements of between the informer and expecto, they are in scale of like around 0.3, 0.4 as well, while it's completely different architecture and much more modern and much more powerful. Here is an example where we have the gene body and some enhancers, and there are some loops that connected, as you can see. And even if the enhancer is quite far from the TSS, it still can be used in our model, which is the reason why it's performing a little bit better. Here we have some examples on the specific cell lines, which you can find in the preprint that I mentioned, as well as the residual analysis and some Venn diagrams that show that we improve in quite a lot of cases the prediction scores. That's all. Thank you for your attention, and if you have any questions, feel free to come by at the ISMB.